Hi, today we're going to be talking about the ultrastructure of cells, and that's section 1.2 in the IB curriculum. There's some things that you want to know. First of all, that the prokaryotic cell structure is much different than that of a eukaryote. The eukaryotic cell structure is in many ways more complex than that of a prokaryote, and the electron microscope has been a valuable tool in kind of evaluating the differences and similarities between these two cells, and it does a lot to reveal the fine structure, or the ultrastructure, the fine detail of these cells. So some of the things that we want to be able to know how to do by the time we're all done is we want to be able to use our knowledge of what we've learned to be able to compose a drawing of an electron micrograph of a bacterial cell and that of each of the two kinds of eukaryotic cells, those being the plant and the animal. And then we also want to be able to look at an electron micrograph of a eukaryotic cell and determine what type of cell it might be or the particular function that it might have. So the electron microscope was originally developed in 1931 to look at non-biological materials. And after scientists kind of got using this, they started to realize that if they could stain biological substances, they might be able to look at and reveal a lot about the fine detail of those as well. And there's two main types that we need to be concerned with. We have the scanning electron micrograph, or the SEM, and the transmission electron micrograph, or the TEM. And the transmission electron micrograph actually looks through the object that you're viewing, the cell and so forth. So if we look here, we can see that we're looking at the polio virus in this instance. And the ability to see viruses is one of the features of the electron microscope that's based on the resolution or the resolving power, the ability to distinguish two objects as separate and distinct objects. And the electron microscope, no matter which type you're using, has about 1,000 times the resolution of a typical light microscope. So you can see down to between 0.1 and 0.2 nanometers. So it's got a lot of resolving power. If you look at here with the scanning electron micrograph, we're going to scan the surface. So these are some pollen grains, and we can reveal a lot about the fine details of the surface of these things as well. So it's got a lot of uses, and it's revealed a lot of very important things about the actual cell. So looking at prokaryotic cells and how these cells differ from those of eukaryotes, we can see that they share some similarities, but they also have some differences in them as well. So both of these organisms, both prokaryotes and eukaryotes, both have a cell membrane, and the cell membranes are slightly different. Their thicknesses are different, things like that. And the similarities that they have are that some of them have cell walls. Some of the eukaryotic cells, like a plant, has a cell wall, whereas nearly all of the bacterial cells have cell walls. Even though they're completely different, they still have a cell wall. Both cells have ribosomes, both cells have genetic material, and both cells in some cases have flagella. In the case of the eukaryote, not all eukaryotic cells have them, and not all bacterial cells have them. So if we look at some of the differences now with them, they're kind of related to some of these similarities. So the DNA, they both have DNA. A eukaryotic cell's DNA is associated with a protein, and it's bound by a membrane, whereas the bacterial cell, it has a nucleoid region. It's kind of naked DNA. It's kind of in a spot in the interior of the cell, but it's not associated with any protein. The ribosomes are different sizes. The bacterial ribosomes are 70S ribosomes. They're a little bit smaller than the eukaryotic ribosomes, which are the 80S ribosomes. The chromosomes in bacterial cells are circular, and they're joined end to end. They're still a double helix, of course. And the eukaryotic DNA is often multiple linear strands, but it's always linear, whereas the bacterial is also always circular. And the flagella, even though they both have flagella, the flagella in a prokaryotic cell is much like a rotor. It kind of spins in a circle, almost like the hook on a bread machine, or the arm on a bread machine, whereas the eukaryotic flagella is more whip-like. So, and that's due to the microtubules that it has. And we said in many ways that the eukaryotic cells are much more complex than the prokaryotic cells. And some of the similarities, again, we talked about these, and some of the differences. The eukaryotic cells are about 10 times larger than that of a typical prokaryotic cell, and they have membrane-bound organelles, and this is what is allowing them to become larger than a typical prokaryote. The genetic material, we said, is associated with histone proteins in eukaryotic cells, whereas it's naked in the bacterial cells. It's not bound with any protein. The chromosomes, again, we said those are linear versus circular, and the flagella are composed of microtubules that you'll see in a 9x2 arrangement, and this is what allows for that whip-like kind of motion that propels these things through the medium versus the rotary type that you would see in a bacterial cell. So one of the things that we wanted to be able to do, based on our knowledge, is compose an electron micrograph of a typical prokaryotic cell. And some of the things that we want in our drawing is we want to have the cell wall, we want to have these pili, flagella, the plasma membrane, the 70S ribosomes, and the nucleoid region. So what we would do is we can draw our cell here, and then we can draw the flagella that we might have here. There might be one, there might be two, but we can draw these, and these things are going to operate again, as we said, in rotary fashion. Okay, so these will be the flagella, and then we might put some pili on there, and these things are often associated with conjugation, which is when they exchange the genetic material between the cells. Sometimes they're involved in just attachment to surfaces, like with biofilms and things like that. So there might be multiple pili on the cell. These cells oftentimes have a cell wall, just depending on what type of cell it is, that's on the outside here. And what I'm drawing right here, I guess you could say, would be on the interior, so this would be the actual cell membrane. So we can label these. We'll call this one here the cell membrane. And then on the outside over here, we would have what's referred to as the cell wall. This is involved in kind of protecting the cell from differences in the osmotic potential on the outside of the cells. We might have a bunch of ribosomes kind of on the interior here. Excuse me. Sorry, the pen is kind of getting out of whack here. So we might have some tiny ribosomes that are associated with protein synthesis, and many times this would be the target of some antibiotic drugs that would kill bacteria if we have an infection. So you can see they're going to be all throughout the cytoplasm here. So these would be, oops, let me go up and undo this. Get back down here. These would be the 70S ribosomes. And then the naked DNA that we would see inside, and this is going to be joined end to end right there. And this would be, we could say, the nucleoid region, which is the DNA of the actual cell. So if you could draw something like that for the electron micrograph, you know, that would be fantastic, and I think that would answer all of the things that you were asked to draw in the drawing here. Okay, so moving on now, if we want to draw the other type of cell, a eukaryotic type of cell, the first one that we might want to draw here would be, say, a plant cell. So the first thing that I would do on this plant cell, they lack flagella, so I would draw the cell wall on the outside of this. So this would be the cell wall. And then in the interior here, I would draw, this could be the plasma membrane. And then here I would draw the nucleus, and of course these are all membrane-bound organelles along with this nucleus here. I'm going to draw the rough ER, and I'll get to that here in just a second. So this, we'll say, is the nucleus. And then the rough ER is going to have these ribosomes studded to the surface of that. And one of the important things to note about the ribosomes is they can switch back and forth between being free and bound at any time. And when they're bound to the rough ER, they're going to be making proteins that are destined for export from the cell. And these we can label this. This would be a bound ADS ribosome. And this is going to be sending proteins off to what we can have here is the Golgi apparatus. Okay, and it's going to be giving vesicles off the end of it. So this we'll say is the Golgi. And these would be vesicles. So we got some vesicles there. And then in addition to this, we would have some free ribosomes. And those would be scattered all throughout the cytoplasm. I'm just going to draw these in one small region here, but they're found everywhere. These are free ribosomes. And what these do is these make proteins that the cell itself uses. And these, again, of course, are just like the bound ones, only they're free. They're both ADS ribosomes. And then one of the other organelles that we need to get in here is we're going to have the mitochondrion. And it's got the squiggly interior here. And this is the cristae. That's the internal. Oops. Go up here. Do this. This would be the cristae. And that's the internal surface membrane, so that's where the ATP is going to be made. And then this would be the outer membrane, but this whole thing here is the mitochondria. Okay, or the mitochondrion, which would be singular. And then you might have this central vacuole. And that's responsible for holding the water and regulating the osmotic balance and perhaps some toxins, things like that. And then in addition to this, we're going to have the chloroplast here. So it's going to have all these little tiny grana in there if we're talking about the electron micrograph, which is what we're drawing here. But this is the site, of course, of photosynthesis. So this is the chloroplast. Oops. This pen is kind of tough to get used to here. That's the chloroplast again. And so I think that pretty much takes care of the thing. We have some smooth ER, I guess, that we could put in there. So the smooth ER, the only difference between the smooth ER and the rough ER, of course, is the attachment of ribosomes. So we can put some of those in there. So this would be the smooth ER. And we didn't label this over there. We said that these are the bound ADS ribosomes. So this is the rough endoplasmic reticulum, or the RER. It doesn't quite look as well as it should here. So this is the rough ER. There, that E looks a little better. So that's the rough ER. And now, if we look at the eukaryotic cell, just a typical animal type of a cell, we could draw the cell membrane here. And we've got the cell membrane here. We've got the nucleus, of course. And then that's, of course, what houses the DNA. We can have, again, mitochondria with all of this internal membrane here. So this is a mitochondria. And then the cristae, of course, is the internal thing like we showed in the other drawing here. We've got associated with the nucleus again, we've got the rough ER, and I'm not going to draw lots of the ribosomes on it, just kind of draw a few of them here. And again, proteins being exported from the cell. So that's the rough ER. We might have over here, again, some smooth ER. Again, the difference between the smooth ER and the rough ER is the fact that it's not studded with any ribosomes, so that's the SER. The smooth endoplasmic reticulum is involved in detoxification, so you find a lot of that, say, in your liver cells and things like that. We've got a lot of bound ribosomes that we said on the rough ER, but you're also going to have these free ribosomes making proteins for the cell there. So we can say that these are the free ribosomes. And then when moving on, and these again, both of the ribosomes for the rough and the smooth, because this is a eukaryotic cell, these are ADS ribosomes. This being an animal cell could have a flagella. Not all animal cells do, but they are got microtubules in them. They in a 9x2 arrangement, they work by a whip-like motion, which propels them through the medium. And then they, because this is an animal cell, they might have some centrioles that are in them. So if we look at this, this is the microtubule organizing center. So this is the centrosome. Centrosome containing the centrioles, and these are going to be involved with the assembly of the spindle apparatus, which is composed of microtubules to help with cell division, and sometimes these are called the microtubule organizing centers. And if we look at this, we've got another organism, or organelle, excuse me, will have a lysosome. This is a bag of enzymes. And a lot of times these things, have a little bit of a kind of a granulated appearance or a darker appearance in them, but these contain enzymes in them. And then if we look at this now, we've got the lysosomes, we've got the centrosome, and these two are very important in or knowing the difference between the plant and an animal. These two are found in the animal cells, whereas if we back up over here, we've got the cell wall, we've got the chloroplast, and we've got the central vacuole. Those are the things that are important in the knowing the difference between the plant and the animal cell. So in the plant cell, we have a cell wall, we have a chloroplast, and we have a central vacuole that is unique to the plant cell. And with the animal cell, we have the centrosome and the lysosome, and these are the ones that are unique to the animal cells. Now, the last thing that we should look at here is just if we're given an electron micrograph and we want to see the difference between these two types of cells. Whoops, excuse me. Sorry about that. Trying to zoom in a little bit here for you. It's very sensitive. There we go. So anyways, what you look at when you're looking at these types of cells, you can see these are basically the vesicles that we have here. And what we have is these are secretory vesicles. And these are going to be containing oftentimes enzymes and things like that. Okay, if we look over here, this is the Golgi apparatus. And this is involved in the packaging and export. So things are going to be made in the nucleus. They're going to travel, the messenger RNA that gets made in the nucleus is going to travel to the ribosomes, and they're going to be directed to the endoplasmic reticulum. So when the ribosome binds, we'll get the rough ER forming, and then the proteins from the rough ER are going to travel to the Golgi apparatus here, and they're going to be modified, tagged, and then these vesicles are going to bud off of the Golgi apparatus and kind of make their way to the cell surface, the plasma membrane, to be exported from the cell. So here, this is the nucleus. And that contains the genetic material, of course. And then this is pretty characteristic. If you're looking at these, you can see all of these ribosomes studying this. This is the rough ER. Okay, so these ribosomes are making proteins for export from the cell, and first they're going to pass through the Golgi, and then once they get to the Golgi, they're going to butt off in these vesicles and then make their way, in this case, to the lumen. So this is a type of secretory cell. Specifically, it's a pancreatic cell. So these are all enzymes that are going to probably find their way to the small intestine during digestion. And then if we look down here, you can see that these organelles that kind of have this characteristic appearance to them, if you remember from looking at cell respiration and some of the images of the mitochondria, these have the specific shape and the internal structure. So these are the mitochondria. So that takes care of being able to identify the different types of organelles within a particular cell, in this case a secretory cell. And I hope you're able to kind of recall some of the things that you learned earlier on in your course. Have a great day.